E poi anche
Okay, so let's start next lecture. So it's a pleasure for us to introduce Manuel Melo from the University of Lisbon. He's expert in coarse-grained modeling, and he's one of the main developers of the Martini field. So thank you for coming, Manuel. So the stage is yours.
And thanks a lot, Jorge, and thanks, Sonsoles, thanks, Inel. Yeah, Sonsoles is there. Thanks a lot. It has been great being here. I think this is a really good initiative. And okay, let's talk about coarse grain. I mean, there's a lot. I was told this is not a beginner's level, so you're going to get the full thing. And let me know if you're not hearing well, because it's not being amplified, okay? And I sat in some of the lectures you already had, so you already heard the word coarse grain at least 3 times that I counted, probably more. So let's get on to it. So coarse grain, here a nice visual example. And first, let's start with a brief recap on MD. There's a little bit at the top there, but this is just a recap on MD and what you need to have MD. So you need a set of degrees of freedom. This is typically defined by the particles, the number of particles that you have that are going to be able to move in space. Then you have a set of boundary conditions, right? The size of the box, and then pressure and such stuff like that. Then you have the force field, basically the set of potentials that govern how your molecules interact intra and intermolecularly. And that constitutes your Hamiltonian, that then from there you can use Newton's equations to get the forces or calculate the accelerations on each of the particles. Particles will have masses, and using that you can know where they're going to be a bit ahead in time. So bringing it all together, you have your MD simulation. Perfect. So you've already also seen plots like this. This is the scope of the models you can use for the sizes and scales, times and size scales of your problems. All right. So if you're doing stuff up to the nanometer and a little bit close to the nanosecond, you can work with pure quantum level models. If you want to go bigger than that, you'll have to go to MD. I'm saying here atomistic MD. So probably to the, let's say, tens of microseconds and tens of nanometers. All right. Just so we are on the same page, everyone knows more or less what these scales mean, right? Everyone knows what a nanometer is. Yeah, everyone knows 10 to the minus 9. Yeah, but so tell me something on the scale of a nanometer. Or 2 or 3 or 4, I don't know. Something in that scale. Like, I don't know, a virus, small virus, one of these bacteriophages. Yeah? That's already multi-nanometer. Okay? That's bigger. No? Some examples? No?
DNA.
DNA.
Like in the nuclear.
Well, yeah, the thickness of it, that's around that. That should be around what, 2 or 3? Yeah. So that's a good example. Another one, for instance, biomembranes. I work with them a lot. They are typically around 4 nanometers thick. Okay, so this is the scale we're talking about. Time scales are harder to exemplify. I mean, processes can take this huge array of scales, but you already heard about folding, for instance, or ligand binding that can go from microseconds to hours, days. So this is what we're talking about. So if we want to go further than this, we need even further simplifications. Okay, so this is where coarse grain comes in, and there's always more. The reason I draw this line here is because, okay, we have to draw a line somewhere, and the way I define coarse grain, or a term that I think it's a good definition, is some models that you sacrifice atomistic detail, but you still can tell molecules apart, right? At some point when you go to these continuum models, you stop knowing what is one molecule, what is the next. Okay, so you approximate even the molecular identity. Here you can still tell what one molecule is and what another one. And there are different types. For instance, okay, you have the octane here I showed in the first slide, and this can be a coarse-grained model of it. It's simplified, 2 particles, but this might be just enough. Okay, we could just as well approximate this molecule by a single particle. Okay, there's even more complex models that you can have a single particle with some sort of anisotropic interaction potentials. Then you could even capture somehow this cigar-shaped behavior if you need that. To be very honest, the computational effort involved in tracking the directionality information for particles, it kind of defeats the purpose of being faster. So it ends up being faster to just have 2 particles in this case. But it's interesting to put there as well. Right? So any questions? If you have any questions, just interrupt me. No problems. Okay. Also, if you can't hear me well. So okay. Now I have a question. Why is this faster? Why is coarse-grained faster than atomistic? Less particles. Good. Right. So that was a given, right? There's more to this answer. I'm going to come back to that later. But yeah, that's good. Okay. So there's a problem now. It's like we are kind of creating particles that they don't really have a very direct reflect in reality, because as far as atomistic MD goes, okay, Born-Oppenheimer approximation and such, you still have a very close connection to what an atom is. But now we're having a particle, like you saw this, had an octane represented by these 2 particles. So you have a particle that represents 4 carbons and hydrogens, another particle representing another 4 carbons. It makes it tough for us to know or to get a basis to how these molecules should be interacting with one another. By example, if I have 2 atoms, atomistic level, I have infrared spectroscopy that tells me like the frequency of the bond between those 2 atoms, and I can translate that exactly into MD and have force constants and bond distances that match experimental data. But in coarse grain, it gets way fuzzier, right? So when you want to create these models, we have to come up with these potentials, this identities, like the interaction identities of these particles. And most of my talk is going to be discussing exactly how this is done. And I'm going to start with the methods that I don't use so much and I don't like so much. Is just a couple of slides. This is what I call structure-based parameterization. And this one in particular is based on the method called Boltzmann inversion. And the idea here is we want to find what is the interparticle potential, the interaction potential, that then reproduces a target RDF. Who knows what an RDF is? Okay, not so many people. Okay, so an RDF is essentially the following. I'm going to exemplify along with the coarse-grained example. Let's say I was working not now with the octane, but with hexadecane. I'm showing the hexadecane there is this gray 16 carbon molecules there. And I have overlaid on top of it what I want my coarse-grained model to look like. So I haven't yet simulated any coarse-grain. I just had my atomistic system. But I just did, for each center of mass of each 4 carbons, I just put one orange bead there. We call them beads to distinguish from atoms. They're just particles in MD, but okay. So each orange bead corresponds to 4 gray atoms. Great. Now, what we're going to do is for each of these atoms, or for each of these beads, we are going to measure the distance to all beats. Okay, so we do an all-to-all distance, and then we do a sort of a histogram of this, and that's the RDF, radial distribution function. All right. So RDFs typically look something like this. They're very useful in a lot of contexts, and the reason why they look like this is because, I mean, this is a solvent. Solvent tends to have structure, and it kind of gets reflected in this RDF. Now, the cool thing is, yeah, questions?
Sorry, is the RDF for each particle or?
No, for all to all. Okay. One thing that we do exclude here, so we don't get the bias, is typically you don't include in this histogram the intramolecular distances, because those are not going to be so dependent on the intermolecular potentials, are going to be based on the bonds. So that's a different thing. So yeah, we just do this intermolecularly, but yeah, it's like that. And this can also be done over time. Doesn't need to be done over a single frame. It can be just done for many frames of a source, a reference atomistic simulation. Okay. So we got this RDF. All right. So through this process called Boltzmann inversion, we can infer what the potential between these two particles, this and this, or these are all the same type of particles. So what the potential of interaction between these kind of particles at a distance, not bonded, at a distance, how it should look like. It's an iterative procedure, but the point is then to arrive at a converged behavior, at a converged potential that when I simulate my coarse-grained system, now we forget about the atomistic, we just put our coarse-grained particles there, or coarse-grained molecules. Now we simulate, and they are going to be subject, their interaction is going to be subject to this potential that I'm testing. And then when the simulation is over, I'm going to measure the RDF of that simulation, like of the particles in that simulation, and see how well it matches my reference one, right? So we just do this, and from the difference, we can know how much we need to adjust this. For instance, this corresponds to the first name shifted. So, okay, there will be a correction to bring this well to the left. This is intuitively explanation, but this can be done in an automatic way, mathematically more well-defined way. And yeah, so when this converges, then you end up with this potential that explains how these molecules should interact with one another. So this is one way of solving this problem of how do you invent potentials for interaction between these invented particles. All right. Before skipping to another type of coarse graining, this is a nice method, but it has some drawbacks. So the nice part is typically the local structures that you parameterize, they are going to be very well kept because you are actively parameterizing to keep local structure in your system. But one thing that you can already see is that, okay, I've done this just for hexadecane. But now let's say I want to put, I don't know, a protein in it. I can't just put a protein there. I have to repeat the entire parameterization procedure with the protein, because then I don't have any potentials that between hexadecane and the protein. So I have to redo it. So anytime I want to add complexity to the system, I have to go through the entire procedure. So that's not so good. So that's probably one of the reasons why these methods are not that much employed. And that's why I also want to get them out of the way right away to move to the ones that I like better. I call them heuristic parameterization, in that the goal is less the reconstruction of the local structure. We will try to keep the structure as much as possible, but then the interactions are parameterized already with an eye to a target macroscopic behavior. All right. So an example is the Martini model. Okay, it's not even the first time you've heard about this in this school, and the reason why people talk about it is, well, maybe it's because it's great, but also it's just because it's used a lot. So there's also an element of popularity here, and the fact that it's popular also means that you have more people working with it, you have more parameters for more molecules, so that there's a sort of a virtuous cycle in using it. So this is a bit different, but how this goes, it's the first part is also involves assigning beads to respective groups in your atomistic reference molecules. So typically, in what was the old Martini 2, you have 4 atoms to 1 particle, to 1 bead. So these are some examples. For instance, proteins would look like this, lipids would look like this, sugars would look like this. But then there are some kind of rules, or at least some criteria that we should follow, because there's a not unique way of doing this. And herein lies a bit of the art, but also some difficulties in this coarse graining, is that there's many ways to kind of place these atoms, these beads over atoms, in a way to construct a coarse-grained model. And so at some point we have to use some chemical intuition, what makes sense where. And one aspect, for instance, is, let's say here we have a phospholipid. This is a head group of phospholipids, and if you're familiar with this is a choline group, this is a phosphate group. All right, so this phosphate group will bear a negative charge, the choline will bear a positive charge. So it's clever, and it makes sense to have these as two separate particles, so actually you can represent each of these charges separately. If somehow we were to have a particle that kind of captured both, that was kind of midway and had a bit of both, that would make it a bit of a in-between behavior, and it would not be so clear in the Coarse-Grained model that there is this zwitterionic behavior there, right? So that is one of the aspects. Also, when you have polar and the polar molecules, or amphiphilic molecules, instead of assigning a mapping that kind of has an average polarity, if you can have apolar stuff next to polar stuff, just like the glycerol here and a acyl chain is there, then this makes it much more clear. You have a much starker contrast, and that will drive your interactions in a clearer way. But this, again, it's a bit of a intuitive attitude. Of course, getting inspiration from other molecules in the force field helps, because people already went through the same problems. So far here in this model, there were no partial charges. The new model already has. So I'm going to show that in a moment. And then, if possible, respect symmetry. Some molecules are symmetric, but sometimes if you want to respect these rules, it's hard to keep symmetry. An example, for instance, before that, be consistent. Okay, so if you're parameterizing a set of molecules and they are all common, they have a common motif, do try to keep the common parts the same instead of just having major differences between those molecules. But talking about symmetry, for instance, if you had a molecule like toluene, and if you wanted to assign some coarse-grained particles to it, be like this 1, 2, 3 atoms, these ones there, this one and this one, but then we kind of break the symmetry. So there was some tricks we could do, like having kind of shared atoms and represent like this. This one would have like these 4 atoms and then 3 and then 3. So this would keep the symmetry. But already you see that there's a problem here, because this is, okay, toluene, it's a ring structure, it's aromatic. But if you want to represent it as a ring, to be a ring, you need at least 3 particles for a triangle. But definitely you're breaking this rule of 4 atoms per particle. And this was a problem with this Martini 2 model, that it was not specifically parameterized to allow for these cases. And that led to some subtle deviations from correct behavior. So the molecules tended to over-interact because of this. So recently in 2021, there was this Martini 3 model. I think there is a title saying Martini 3. I think it's one of this part here that is being cut by the ceiling. Anyway, so this is the newer model, and now the strategy is a bit different. So now you can also have 4-to-1, but also 3-to-1 and 2-to-1. So this makes life much easier when you're dealing with aromatic stuff and such. Now the target is also there's a new target. So one of the objectives is to match the reference volumes of your target molecules. So you have this surface accessible solvent accessible surface area. So this is the part that is the limit of what is accessible by a solvent molecule due to the van der Waals radii of the atoms. And so the idea is to have sort of a match between what your coarse-grained solvent accessible area and the atomistic one is. This is an example we had recently. We parameterized the phosphoinositides. This is a head group of the phosphoinositides. And then this mesh represents the surface. The blue mesh is the atomistic surface. The orange mesh is the coarse-grained one. So the particle placement was now not center of mass. Now it was just placed to try and, as much as possible, cover the same thing. You can see it's not perfect, and one of the reasons it's not perfect is because this surface is going to be dictated by the atom shapes, and there's many more atoms atomistically, so it's a bit more rugged. But in coarse grain, it's going to be smoother. But in the end, we were happy with this, and and this new strategy really allowed this new model to overcome these limitations that are this kind of bugs or not so correct interactions that were before. But there's still the question. Okay, so sure, but so now there is this new mapping scheme, but still, how do you make these things interact? We saw with the Boltzmann inversion how you could find those potentials in that case, but in this case, in the Martini, how do you decide how these things interact? And as you've seen in the MD lecture, so you have the bonded potentials and you have the non-bonded potentials. Okay, bonded between intramolecular, non-bonded at a distance. So these ones, we want to be potentials that recreate locally the configuration that the molecule should adopt. The non-bonded, and this is where this target, macroscopic target, comes in, is that we want these molecules to behave in a way where they display the correct preference for polar and apolar environments. So we call this the partitioning behavior. So maybe some of you have heard about partition coefficients between octanol and water. Some of you have heard about it. Yeah, some say yes, some say no. This is essentially the case. Okay, when you have the separation funnel and you have an aqueous phase in the bottom, an apolar phase on top, typically water in the bottom, octanol on top, and you put a solid in there, shake, and depending on its polarity, it's going to prefer to be in one phase or the other, but it's almost never 100%. Or it's never 100%. It's you're gonna have a partition, and from that ratio, you get your partition coefficient, and it's a sort of a reflex of how polar or apolar your molecule is. So this is the kind of target that we have for Martini molecules. It's one of the targets we have, and you might ask, so why are we looking at this? I mean, yeah, anyone wants to venture why we want our Martini molecules to represent good octanol partitioning data? Seems a bit dry objective, no?
Well, lipophilicity explains a lot of biological properties.
Oh, there you go. Yeah, so life is just fat, right? So and then fat and non-fat, and then there's some charge. Basically, that's it. So if you get this right, we're getting half the way to doing biomolecular simulations, right? So that's it. That's exactly it. So there's lots of experimental partition data because it's very important in a lot of pharmacological predictions. So there's an interest in having those data. So that's great. And for instance, if we want to use experimental data also for the bonded part, there's some specific examples where you have a lot of information for there as well. So I'm showing here the example of proteins. For instance, when this model, this was for Martini 2, but the example stands. When it was being developed, for instance, at some point, we had to decide, or it had to be decided, what was the distance between these two backbone beads. Okay, these represent the backbone. And so there's this huge database called the PDB, the Protein Data Bank, that has a lot of experimentally determined protein structures. So you could just map those structures into coarse-grained and measure, okay, what's the typical distance between backbone particles? And it turns out that it depends on secondary structure. If you have this mapping here like this, the distance depends. It's shorter if it's an alpha helix. It's longer if it's a beta sheet. It's in between if it's coil state. So based on this, it was decided that I think in the end the parameters depend on secondary structure. So if you have an alpha helix, they're going to be shorter. If you have a sheet, they're going to be longer. But these are going to be the distances that are going to be employed in the coarse-grained model, okay, directly from experiment. Okay? No atomistic simulations. Great. Now, but sometimes you don't have experiments for either this or this. So now, yeah, we have to turn to finer examples. For instance, if you can get atomistic model for your molecule, you can simulate your model atomistically. You map your coarse-grained beads onto that atomistic model, and then I'm going to be sharing the slides, by the way. So videos and everything, so don't worry. So you you map your coarse-grained beads onto the atomistic molecules, and then you measure how those bonds behave, how those distances behave over your trajectory, and you get something like this. Let's say that this histogram here is how the distance behaves atomistically, and then you just trial and error, you do it in coarse grain. Well, I think this bond should be 4 angstrom long with this force constant, and you see how it goes. Maybe it's too loose, maybe it's too tight, and you adjust till you get agreement. All right, I'm going to show that example later. Now, for log P's, you can also get them from simulation, and actually you talked about that yesterday. So this is one of those examples from free energy calculations where you can actually use this alchemical transformation to get partitioning data out of simulations. And I'm going to show that in a slide a bit ahead. So before I show you the example of the alchemical transformation, just a bit more on the bonded potentials. So which are we going to be used? Which are we're going to use? And typically in Martini we stick with the ones that are available in the GROMACS software, because Martini was kind of developed with GROMACS essentially. So it's a bias for that. Not to say that other things are not possible, but that's how development has progressed. So bonds are the typical quadratic potential, nothing special there. The angle, they use this cosine square instead of just the difference of the degree square, and the reason has to do with some possible numerical instability when the angle becomes collinear, but it's more or less your typical angle, but it behaves a bit like this. This is the cosine squared one. Yeah, there is one aspect of it is that it becomes weaker towards 180. So, like if you want to keep your angle at here, you have to bear in mind that it's the force keeping it to going to 180 is not going to be as strong as the one bringing it to 90, for instance. And then the dihedral angles. The dihedral is a torsion, right? So, and there's two types of torsions you might want to keep in your system. One is really the periodic torsion that really corresponds to a bond that can just go around itself several times, like rotate around there. And the other ones is like in rings or stuff that should be actually always more or less in the same orientation, that should not actually flip over. And for that, you have two different potentials. This is the periodic one, so they can keep on rotating. This is the one that should only have one minimum, and these are their shapes. Now, this is pretty standard, and probably you heard about this in the MD talk, so nothing special here. What is interesting here is when you start working with coarse grain, we're living in a mystic world in the sense that GROMACS and most of the engines that we use, they are optimized for atomistic, and they're prepared for atomistic, and all these potentials, they were first developed to represent atomistic molecules. Fine with that. Problem is that there are some subtle problems that crop up mostly with coarse grain that these softwares didn't have to deal with. And one of the examples is when you're computing the force that is generated by distortions, at some point, there's a derivative there that involves the angle between the particles that you are measuring the dihedral over. So one thing is the torsion angle, but now we're talking about this angle, just the bending angle here. So if this bending angle or this bending angle become collinear, there's a term that crops up in 1 over sine of this angle, and that will diverge to infinity because sinus goes to 0 because sinus of 180 is 0. So the force is going to diverge. So if you get close, it doesn't need to be a 180. If you get close to 180 degrees, you're going to get numerical instabilities. And typically, this is not a problem with atomistic simulations because the bending ranges of a set of atoms, they are very narrow. I mean, the water or like 109 degrees. I mean, it will vary like 5 degrees either way. It's not going to be able to do this. It's never going to be collinear, right? So this is not a problem that would affect atomistic systems, but it does affect coarse-grained systems because now we're talking about usually much smoother potentials. So it's possible for molecules to become collinear. And then if you have a dihedral potential on top of it, it's going to be problematic. The solution, one of the solutions, there's several, but one of the one that I use more often is now going back to the angle potentials, so the ones keeping these angles as we should, instead of using either the red or the black one, using a new one called the restricted bending that has this shape. So that goes to infinity at 180. So it really has a strong barrier keeping your system from becoming collinear. So basically, you're just putting a stronger force, preventing that unstable case. All right. All right. So just to recap this part, the typical Martini approach is you obtain your target distribution from either from experiment or from your underlying atomistic system. And for that, you map your particles onto your beads onto that system, and then you measure your angles and the resulting bonds and such. Then you assign one of these potentials. I mean, depending how you're doing this can be done more or less automatically. If it's a simple case, you can do it by hand. If it's thousands of molecules, you'll have to find a bit more of a streamlined way. Then you simulate that coarse-grained model, and you compare the resulting bond angle distributions to the target ones, and you repeat it till you're happy with this. Okay. This is an example. For instance, I was working with this model with this molecule, and at some point I was looking at these two beads there, and I need to know, okay, what's the distance I'm going to put here, and what's the force constant I'm going to put in this bond. So atomistically, this is the shape it has. And my first try was this a bond with the 0.415 nanometers and this force constant. But then I simulate, and the distribution of what comes out looks like this. So what happens here? You can see it's a little bit shifted to the left, and also it's a bit spread out, too spread out. Being too spread out means that the force is not tight enough, right? You can see if I increase the force constant, this is going to be narrower. All right, second try, same distance, stronger force, getting better. And then I think the last one, I increase a bit the distance and the force as well. And at this point, I was happy with it. All right. So there might be a little bit mismatch in the representation of how it behaves here. But I don't think this is going to really affect the overall behavior of the molecule, right? Repeat this for all the bead pairs, all the bonds and angles and such, and you have the coarse-grained model. All right. Now there's still on this bonus part. There's lots of tips and tricks. Sometimes you have this highly localized distribution. Let's say you want to do benzene and you have these three particles. And I mean, I don't even need to run the simulation to know that this is going to be a very, very well-defined distance because this is all aromatic. So, but yeah, okay, just for fun, you simulate, and then you come up to a distribution based on atomistic model that is very narrow. Okay, sure. So I just said, if you want to have a narrow distribution, you increase the force, right? But if you want to have an almost infinitely narrow distribution, you're going to need an almost infinite force, and that is a problem. So for this case, what we do is to use constraints. Okay, I don't know if you talked about constraints in the MD lecture. No. So using constraints is a way of solving Newton's equations of motion, but specifically saying that these two particles are exactly always at that distance. So it's done in a different way. There's here a schematic of the algorithm that's used with GROMACS, but in that way you avoid actually having to set a force. Like the equations of motion are going to be solved exactly keeping that distance like that, and that's going to save us problems by the problems of using high forces. I'm going to show later what those problems might be. Now, other tips and tricks. If you're ever parameterizing a molecule, don't over-restrain bond interactions. If you've ever done stuff like this atomistically, you'll notice that all angles, all torsions, they're all defined. In coarse-grained, it's the opposite. The less is better. And the reason here is that you're trying to complement the system with whatever its own internal rules already can't do for you. So if my molecule already behaves well just because of its own internal repulsion and such, just having some bonds, it's fine. I don't need to be forcing it with an extra angle if it's already behaving well. Often that might be problematic. Let's see an example. Let's say I have a linear molecule that has some aromatics at either end, and it likes to do this. It likes to stack because aromatics. Great, right? So if I do my atomistic simulation, there's going to be a bias for this configuration. And the central part of my molecule is going to have a sort of a narrow bending angle because it's representing this stacked case. Fine. And I want to parameterize this molecule, and I see this narrow angle. So I say, okay, I'm going to put a potential there, keeping this angle narrow. But now let's say that I'm simulating this in benzene, which is also an aromatic solvent, and might interfere with this stacking, might actually solvate this and break this, and my molecule would open. If I have parameterized this to be a narrow angle, my molecule will never behave correctly because now I've over-parameterized it to a specific situation. But the thing is, if I had in my initial parameterization, if the thing already worked like that because the aromatics were interacting, I don't need to reinforce that. And actually, reinforcing it then prevents correct behavior when the conditions change. So less is best. Also, another thing to take into account, and this bites people every time they start parameterizing this kind of molecules, is sometimes you are not aware that particles might be too close together. Let's see this example. I'm going to zoom in because it's so this is the sugar head group of a ganglioside, but essentially there's a sugar here, and we were looking into parameterizing this connection between these two rings, just one triangle, another triangle. And the thing is, it was not obvious, but this particle was getting too close to this particle, or it gets too close to this particle in the atomistic simulation. And what do I mean too close? The particle types that we were using in coarse grain, they start repelling each other around this distance here. So in my coarse grain system, when I have these particles, and if I don't take care of this distance here, what's going to happen is that they are too bulky. They are going to be repelling each other. And then what will happen is I want to keep this angle close to 80 or 90 degrees as it is, but the system won't let it because they're actually repelling. So it's going to be higher than that. And I don't understand why it's not working. So I increase the force of the angle, really trying to close it, but the particles are just repelling. We're in this repulsion part of the Lennard-Jones potential. So you end up with a super strong angle, keeping it there, and the particles just fighting against it, and it's a recipe for a disaster. And it's just because we didn't notice that, okay, the particles were getting together. Once you know that, either we can exclude them, so you can tell the software to just not count this interaction, so there's no more repulsion, or we can actually put a bond there. I mean, the bonds don't have to follow the chemical connectivity only. Bonds are just harmonic restraints. So we can put a restraint there, keeping it the distance to the distance that it should have. So yeah, this is more advanced tips and tricks. All right, so now for the non-bonded parts. Okay, so in Martini in specific, when you want to decide how these things are going to interact in a non-bonded way, so we resort to this kind of Bible. It's a sort of a list of typical chemical groups that should have typical what's called particle types. And each particle type essentially encodes a specific type of Lennard-Jones potential. Or in other words, not more exactly, each particle type in conjunction with another type is related to a potential, meaning when this particle meets this particle there, this is going to be one of these types, the other one is going to be another type, and there is a table that lists, okay, all these these pairs of interactions, each has this specific Lennard-Jones potential. So essentially it's that. This simplifies a lot because if you can have this building block approach that we just look at, where we just look at the chemical nature of each block, yeah, it's as easy as looking stuff up. So in this case, for instance, yeah, this bead here was assigned bead type C2, which is a branched alkane, and this one is a diol, so it was a type P4, right? And then in the backstage, that's already been in the development of Martini. These kind of guys are going to be interacting preferably with other alkane stuff and apolar stuff, and this is going to be interacting more with water and other polar stuff. And there you go. That's how it works. Of course, you have a larger gradient. It's not just polar and the polar. There's a gradient of polarity and behavior there. But overall, this is the gist of it. All right. There's some leeway. There's some cases where it's going to be like, is it this type or that type or that type? Then there are several kind of rules and cases or adjustments you can make. The Martini paper, the Martini 3 paper lists all these guidelines. So I'm just showing this image here. So there's fluxograms and flow charts that you should follow and such. So it's pretty well explained there. Now, going back to what I mentioned earlier, how do we get a partition-free energy from simulations? And okay, okay, so why are we talking about, again, partition free energies? Is because after we've assigned all our molecule, now we have the bead types for all our beads, all right? So everything has been assigned. I want to make sure that is consistent with target behavior. So now, because we're not just talking about one particle, now we're talking about the entire molecule. And this is where the overlap with the free energy calculation lecture comes in. Basically, what we want to get is this free energy of partition from our solid in water to our solid in an apolar phase, typically octanol. Cool thing is actually if you simulate it in coarse grain, like cyclohexane or octanol with water, they phase separate very neatly. Okay, so you can do this. So you could actually do the simulation really just like a separation funnel, where you put your solid there and let it go. And instead of counting how much is on top and how much is on the bottom, you simply count how much time it spends on one phase, how much time it spends on the other phase. This could work, and it has actually been done. But there's significant problems with that. Because first you have to get significant counts in both phases, right? But if something is very hydrophilic, for instance, you might never see it in the other phase. It will go there at some point for a small amount of time, but you need to have converged value for that, and otherwise you'll just get infinities or zeros, and that is going to be problematic. And then another one is that interface may play a role. So you want to see if it's here or here. What if it spends most of the time here? Then you don't know, is it water or is it octanol? And then you discard those frames and it's just waste. And definitely it's too expensive if you want to use it atomistically, right? Because you can still think about doing this with coarse grain, but atomistically it's too expensive. So what we do is we use alchemical transformation to compute these free energies. And so the idea here, you have your solid in water, and you get to get this free energy of this transference here. And we're going to decompose this in solid in water to solid in vacuum, and then solid in vacuum to solid in octanol. Okay, they have this thermodynamic triangle. If you get this plus this energy, it should be the same as this one. So essentially, it's the same as asking this question here, saying that this is water and this is octanol, and what's this energy here. So this is too big a difference in the systems are too different to get this free energy directly by endpoint methods. But we can then do our chemical transformation, and the way it's done is we have our solid in water, and then you have another system where we have the same solid in water. Now the interactions are just at 90% or so, or 60, or 20, or 0%, right? And the cool thing here is that this system here, where we have our solid not interacting with water, it's just like having our solid in vacuum. So this, in the end, is analogous to this system, because our solid is already what's called decoupled from the solvent. And because now the differences in phase space visited by each of these states are much smaller, we can sum over these small consecutive delta G's to get an overall delta G. So that's how we get the free energy from simulations. And it's almost more efficient than running a system with separate phases. How is this going? All right, still. Great. There's a bit more to it. So this is not your...
This might be just my lack of knowledge of field, but why would you need to do the chemical transformation instead of just using the initial and final state to have the delta G? So
so it depends really on the methods that you're using to compute this. And so the method, I'm going to try and explain in an intuitive way. It tries to measure how often is this state in a configuration that actually would be low energy if you were considering it to be like this, and vice versa. How often is this state in a configuration that is low energy if it were like this? Before I go in deeper, this change here. Okay, we get the solid to the vacuum. This is not the whole story, right? So what did we do with the solvent? So the solvent has to be there somewhere. That's why this is actually a better description. Solvent is there, the solvent is still there, but let's assume that this system was actually this one here. Like, okay, we have the solid completely decoupled, just floating around the solvent. And if I take any like a structure like this configuration here, and evaluate it with this Hamiltonian, so with the settings of this state, would this be a low energy? No, not really, because you have this overlap between solvent and solid. So this energy would be very high. Okay, so this would not really allow you to compute easily an energy. And the other way around, so would this state actually be ever occurring in this Hamiltonian? Well, if you think about it, like the solid here is not interacting. So this would mean that the solvent would actually have created spontaneously a cavity that for itself is just full of vacuum because it's not seeing the solute. So it's also very rare to happen. So basically you're saying, how often does this very rare event happens on the other one, which is never over never? So basically you have a never over never situation. But now if you do it very softly, so from here to here, there's going to be a lot of cases where this state, this kind of state is in a configuration that's compatible with the previous one and vice versa. So you'll be able to it's not going to be never over never. It's going to be now you're going to get some confidence in your in number and be able to compute it very intuitively. Okay, so that's the overall. Questions? No. Okay, so beyond just this specific this more generic target, for instance, we can tailor this to our applications. For instance, biomolecular applications, as you mentioned correctly, we are interested in this hydrophobic and hydrophilic dichotomy, but it doesn't have to be exclusively partitions. For instance, when side chains of amino acids were being developed, because the interactions with membrane is an important objective to look at, the profiles of how these side chains interacted with membranes along the distance, they were measured atomistically, and then this also became a sort of a parameterization target. You wanted your side chains to have the bead types and configuration that when you drag them across the membrane, for instance, they behave energetic-wise as the atomistic reference does. Okay, so this gives you more of certainty that each amino acid is going to prefer to be at more or less a correct depth in the membrane, and then if you have a peptide or a protein, then the overall interaction is going to be correct. For instance, when that ganglioside head group was being developed, one thing that was looked at is how these things actually interact with one another. These gangliosides, they do interact with one another, they can aggregate, and that aggregation is not random. Some parts of these rings prefer some other parts of the rings. So we can measure that atomistically and ensure that what we assign, the types that we assign to our coarse-grained model, then end up reproducing more or less that preference. Okay, all right. So if we have that sort of target data, that is also something good to include. Now, this can be a lot of trouble, and recently people have been trying, well, already for a while, but more and more to automate this process. My opinion is that this is good at the stage where if you need to do a lot of molecules, then you probably have no other option. I think there's still a lot of hand tweaking that for my taste, I would need to have in developing a new molecule. Some of these automation processes, they even just deal only with the bonded part. They don't deal with the deciding what gets mapped where. So that's something that you would still have to do yourself. Also, there's the problem of getting the source data, either it's atomistic reference or experimental. So we are not exactly there yet when you say, okay, I have a library of 10,000 molecules, let's just automatically parameterize this and see how it interacts with my system. So, but hopefully soon. I mean, there's the buzzword AI. I guess AI is going to do this at some point, but not yet. All right. So the overall of this heuristic parameterization of Martini, good things. It's broadly applicable. It's transferable. This is a property that I mentioned earlier that these other models didn't have, that I parameterize this molecule within this philosophy of Martini. I parameterize another molecule separately within the same philosophy. If I bring them together, I'm more or less confident that they're probably going to play well together because I followed these overarching rules, right? I don't have to reparameterize them just for the sake of bringing them together. So that means less work. Typically easy to use, although I guess that depends a bit on the user preferences. We're going to see now in the afternoon tutorial what you think. And the thermodynamic properties tend to be well kept because, well, especially this partitioning behavior, because it's one of the targets. All right. What's also good? Well, as with any coarse graining strategy, it's typically semi-quantitative, meaning that you're most likely to get correct trends rather than absolute values. You're going to lose chemical fine detail. I mean, this goes without saying, right? So if you're looking at very specific chiral behaviors at the moiety level, it's going to be a problem, right? If you have one particle, one bead representing a chiral group, and yeah, a bead that doesn't have up, down, left, or right, so you're not going to be able to represent chirality, let's say. Or if you have very fine H bonding patterns, I mean, it's one particle. The particle doesn't have directionality for H bonding and anything, so that's not going to work so well. And yeah, so this might lead to suboptimal structural features. Now, the thing that I just said, especially the H bonding, is very important for proteins, right? So if you want to have protein structure, you do need to have H bonding between the carbonyls and amides of the backbone, right? So Martini proteins can fold. So, okay, so yeah, let's just do lipids and let's forget about the proteins. Well, no, there's exactly what we use is just we use bonded restraints. So not able to fold means we can't use Martini to go from a stretched sequence to a folded protein. But if we know what structure it's supposed to have, because there's the PDB and people have determined it experimentally, we can actually impose that with restraints. And this is called an elastic network, for instance. So we just keep each backbone at a certain distance from its neighbors, and overall this will keep the shape of a protein. Of course, the protein will not unfold. So if you're looking into that, the flak, okay, but it will behave with its shape more or less. So that's networks. And if you just need local motifs like helices, you don't even need these elastic networks, because for helices, for instance, it's enough to just have torsions and angles. And with the right torsion and the right angle, you just define a helix, and it's always going to be a helix, right? But at least you can simulate it as a helix without just having a jumble of Martini particles. Now, there might be a more detailed future. There's efforts now going in the direction of having multiple particle backbone representations. So this might allow already to have some explicit H bonding behavior. So who knows? In the near future, maybe we have some degree of foldability. That would be great. All right, so this is what I just recapped. And as for Martini, okay, so we're representing typically 4 atoms as a bead. Now we can do less than that. So you have fewer particles, as you said, plus a smoother interaction landscape that leads to a longer time step. And so we're typically looking at speedups between 100 and 1,000 times compared to atomistic model, right? So this is why we put up with the not so good parts, is because, yeah, the scale that we're talking about are completely different now, right? And you're going to see them in the application. Now, earlier when I asked why the coarse-grained models were faster, you said because, okay, you have fewer particles. But there's another aspect here, is that because this potentials now are representing sort of an average of several behavior or interactions between atoms. This kind of averages out, and the potentials tend to have lower frequency or lower force constants than atomicity, and that allows us to use a longer time step, right? And I don't know if it was clear from the MD talk, but essentially it's like this: if you have let's say, a sharp potential, high force constant, and this is your particle and it's feeling this harmonic potential. Okay, you're doing your simulation, and you have to integrate it in a way that it kind of feels the potential, gets accelerated, and then it breaks and it gets back, and this particle will just be here forever oscillating. All right, but now this imposes sort of a limit on your time step of your integration, right? Because if we do the same thing, but now with a faster time step, what's going to happen is that the system might actually miss the breaking, and actually you end up with more energy than you started with, okay? So this can be problematic for the particle. Now, if your potential is much smoother, you can see you can have a large time step, yeah, and everyone comes out alive. So this is one of the reasons why you can use a longer time step in coarse grain, and it directly gives you much more performance, right? Just typically it's 10 times faster, the longer the time step than an atomistic. So you just get 10 times performance just because of this. All right. So now some examples of multiscale. I'm not going to spend too much time here. So especially for those of you who've worked with QMMM and such, there's models to kind of combine the two resolutions spaces. This would be great to do with coarse grain and atomistic, so you could have best of both worlds. I mean, there's stuff a bit like this that has been done. Essentially, you have to have a atomistic region and a coarse-grained region. This is extremely hard and cumbersome to implement, and the big problem is that you have to have this kind of a in-between layer that has mixed or a hybrid behavior. So that's really not the best. And overall, for all the work you have, you typically don't really get any performance speed up just doing everything atomistic. So it's more of a theoretical possibility right now than a reality. You have another alternative, it's called the hybrid scheme, where you actually simulate atomistic stuff in the middle of coarse grain. And how this would work is that you have some, let's say, virtual coarse grain particles that are mapped on top of the atomistic ones. And so the interaction with the coarse grain world is done via these virtual atomistic beads. But the interactions with itself, atomistic with atomistic, would be at the atomistic level. So that would allow protein folding to occur and such. So that sounds great. The main problem with this is that the atomistic models have a lot of charges and partial charges, and the coarse-grained world has almost none. So these charges would kind of exist in a sort of a charge vacuum, and they are not meant to exist in a charge vacuum. These things are parameterized where almost any neighboring molecule has charge. So suddenly there's a charge vacuum, which means that any opposing charge are just going to stick together, and that's a very problematic thing. The behavior becomes very non-realistic. So yeah, so this is those examples. Now there's a third type of multiscaling that's more in time, which is okay. Yeah, we have our atomistic system. We can go coarse grain, but what if we want to go from coarse grain to atomistic? So this would be multiscale that you simulate atomistically, and at some point you just convert to you simulate coarse grain, and at some point you convert to atomistic. And yeah, there's a type of model that does this kind of things. Essentially, you place the atomistic atom semi-randomly in each corresponding to each bead. So I know that these 4 atoms correspond to this bead, so I put them in this region. I know that this atom and this atom are connected, so I can already displace them a bit more in the right direction and such. But this is still not a really correct representation of this lipid. It's just the atoms are more or less placed where they should. Right? But then we can just do an enantiomerization, and yeah, and then suddenly you have a workable lipid model, right? And I mean, this is for a single lipid, but you could do this for an entire membrane. Okay, so yeah, this backmapping strategy works. There's others coming up. Yeah, it's also a field in progress, but yeah, there's some things to bear in mind there. Okay. So now let's look at some applications. And the cool thing now is that we have some videos. I mean, I already showed some videos, but I mean, real ones. So the beginning of the Martini model was very successful because since the lipid interactions are very much dependent on this amphiphilic behavior, what's polar, what's not polar, membranes could be modeled in a very successful way, either as vesicles here of different sizes, or even as just plain bilayers, but then you could already see phase transitions, which is actually pretty cool to simulate. Then there's a bit more advanced stuff. This is actually connected to experimental non-behavior, is that when you have a membrane that has a saturated lipid, so saturated lipids are more ordered, right? The tails tend to be straighter. So you have a saturated lipid, an insaturated lipid, or unsaturated lipid, and cholesterol. These, depending on the concentrations, this might split into 2 phases: an ordered phase that has cholesterol and a saturated lipid, and another one with the unsaturated lipid. And this actually might be connected to what's called the raft hypothesis. I don't know if you've heard about it. It's an idea that in our cells, in our cell membranes, there might be regions that have more order or less order, and that would drive the localization of some enzymes, some metabolites, and it would be a sort of compartmentalization. The jury is still out on that, but yeah, so just an example, we could simulate this. And what I'm showing here is the top view of a membrane. The green is the saturated lipid. I'm just showing the 1 bead per tail. That's why they come in pairs like this. This is the unsaturated one, the red saturated one, and cholesterol is in blue, just one bead. Okay, I'm just showing one leaflet from the top. They start randomly, and if we start simulating very quickly, they split into this emergent behavior of this phase separation. So this is pretty cool and matches pretty well what happens in the lab. But now in the lab, there's a question, okay, what does this really happen in our cell membranes? Because it's one thing is to do it with 3 components in a controlled way with fluorescent probes, but in the cell membranes, it's way noisier and noisier. So 10 years ago, we tried to do this, and this was a record back then. So we did an 80 by 80 membrane with more than 60 different lipids, and it ran for over 80 microseconds. So this took 1 year. Yeah, so this was Helgi Ingolfsson's work back in 2014, and I was also in there. This was a huge work, and honestly, for all this work, it was not very clear the answer. So it's a very complex system, and there was no very clear separation, but there was some preference for some lipids to hang out with other lipids that tended to also be saturated or unsaturated. So there were some preferences, but well, jury's still out, and hopefully in the future with more simulations we're gonna get there. Now, with some proteins, this is just one example amongst many. This is bacterial membrane pressure escape valve. Let's say you're a bacterium and someone just throws distilled water on you because of osmolarity. You start sucking in water, and at some point you just might burst. So if you're a bacterium, you rather just open valves and let stuff out, metabolites, than just dying, right? So this is the protein that does it. And what it detects reacts to a membrane thinning. So this is a membrane, and when the membrane gets stretched, it gets thinner as well. Right? You can see the pressure will increase the area of the membrane, and by response, it's going to get thinner. And in response to that thinning, it's kind of loses the tertiary structure and opens a pore. And then we can do that. We could see that with Martini. And then if you release the pressure, it will go back to what it was before. So it was very cool. This was really, really cool. And you could even have the example here in this paper where, okay, it opens and starts leaking solvent out. But if you don't have that, at some point with the pressure, it just breaks this vesicle and just basically kill the bacterium. All right. Now, this is another work I was involved in, and this is pretty cool. So we had these collaborators that say, oh, we have this mitochondrial protein, or have this lipid, which is ceramide, and if we put it in the mitochondrial membrane, it drives up apoptosis. Okay, it's a good thing to know. But they said, oh, but we found out that this interacts with this protein partner, but we just don't know how it interacts. So can we tell us anything about that? So what I'm going to show you here is a movie. This protein actually turns out to be a membrane channel, but since we're talking about a lipid, the interaction is not going to be so much through the channel. It's going to be the surrounding part. This is top view, but we're going to see it sideways, and I'm just showing the specific lipid. So we simulate, and whoop, yeah, binding site, right? So it's a very obvious interaction with the binding site. So it stays there for a while, and it's quite neat. You can see it there. Yeah. And at some point it leaves. Good. So we could find the binding site. We even found this lipase activity that kind of this protein promotes the lipid from flipping from one side to the other. But in this particular case, I think just show you 2 microseconds out of a simulation that went to 120 something microseconds. So in the total, over the several replicas, we saw over 1,300 binding and unbinding events, right? So then we could even do statistics on how the residence time of this binding and unbinding, and plus these super cool movies, you know? And I was thinking earlier, okay, why do I think that coarse-grained movies are cooler than atomistic ones? And I was thinking back in the conversation 2 evenings ago. And I thought that it was, okay, yes, it's because you can use these balls, and the balls seem squishier, and it's more fun. But no, you can also do that with atomistic if you want. But I think this is the time scales. Now I realize time scales because atomistically, you usually don't reach a time scale where you see molecules floating and interacting with one another in that scale. Mostly you're going to see small behaviors, intermolecular rearrangements and such that are not so visual like this. Okay, so another example, for instance, here is a cofactor binding. This is a bacterial membrane, and there's this respiratory cofactor, and this enzyme is supposed to interact with this cofactor. Okay, there is some interaction there, but the problem is that the binding site is actually here where the laser is. And what's interesting is that you can see that over time, this enzyme actually catalyzes the pulling of that head group. You can see the head group already poking out. And if we wait a bit more, it's going to become more evident. Yeah. It's a longer one. But the thing is, if you don't have this enzyme and you just measure the energy of pulling these lipids out, they don't really like at all to come out of the membrane. But here we see, well, the head group is out, right? And if you do this without enzyme, no way this lipid is ever going to stick this far. So there is a sort, besides the catalysis itself, which is would be electron transfer, which of course we're not doing with coarse grain. There's the catalyzing of pulling this lipid out. Now this is something that's super cool. Is a current project we have. So we're studying this polymer. It's suberin. This is kind of, it's a sort of an average chemistry, but basically this has this kind of glycerol linkers and then this stuff that looks a bit like lipids, but they also have some polarity there. And so when we make our molecules, they look like this. They immediately clump into a little bit of an antipathic little ball, and then we've got them to play together, and they will just start coalescing because they have still quite a lipidic nature. So if you pay attention, you'll see some of them coming together. I think these two here now are going to slurp each other. Yeah, there we go. Those two, yeah. So in the end, if we wait long enough, we went up with a large particle. This is if I cut it, so you can see the color is different. So apolar is blue, and pink is polar, and red is charged. So okay, the center is more apolar. It's not completely apolar, but it's more apolar. All right. So now this is known to have antimicrobial properties. So, and we suspect it has to do with the bacterial membrane. So we just put this next to a bacterial membrane and see what happens. And we're not biasing this in any way. That's why we have to wait for it to diffuse and randomly find the membrane. Okay. Bear with me. So at some point it goes there, it goes there, it goes there, it goes there, and bam. So you start having this very visual mode of action that it hadn't been described yet. It's super interesting, and then you can also see that there are some hydrophilicity events going here. The hydrophobic part tends to stay more in the center of the membrane. Makes sense. So if I were a bacterium, probably I would not be happy with this. And we also can see some increased leakage next to this. So yeah, so this is a cool project, and there is tons of other systems. We had a review a couple of years ago where I did I was counting what were the most components in a given system. So we started small back in 2007, like with only 3 or 4 systems before 3 or 4 components beyond the solvent, and then more and then more and more. This was that big lipid membrane paper, and then you start having proteins. So now you're, yeah, well, maybe you are going to be the next record holders here in this step. And then people have brought this to the mesoscale. So this is a mitochondrion size simulation. It's a multi-scale in the sense that solvent is not being explicitly simulated in between, but the membrane is being explicitly simulated. So this is work by Vuria, and it's pretty interesting. So it shows that you can still use you can even use this to go into larger scales. So this is the end of the first parts of my talk, and maybe just take 5 minutes to think. Maybe questions? No? Yeah.
How do you account for like the electrostatic potential of the solvent in these simulations?
Yeah. So good question. Not specific. So water does have, of course, a very obvious dipole. There have been some models that try to include that, but what Martini does essentially, first, one water is coarse-grained, one water particle is coarse-grained to represent 4 waters in reality. Actually, most of the speed up comes from there because most of the systems are using water. So if you just 12 atoms that are in 4 waters, this become 1 particle, that's being safe. Those are encoded in the Lagrange potentials, right? So if you're a polar particle type, you're going to hang out more with water, and implicitly you're saying, yeah, you're polar because you have some dipoles. Water has dipoles, you hang out together, right? So that works like that. If you have formal charges, of course, water would be screening those charges. So you have this dielectric effect of water. And for that reason, the electrostatic in Martini, they are scaled down by 15 times. So there's an implicit electrostatic, an epsilon of 15, right? Now, yeah, this works. Of course, it's not perfect. And one of the problems specifically is, yeah, I have a dipole. There's another dipole there. Water is also dipolar. We each like each other. Great. But those things will all be given together. Now, in some of dipole interactions, actually what you have is when you establish, let's say, an H bond or a salt bridge, those interactions, they become kind of satisfied and become less visible to the outside. Like if you have complementary charges already kind of screening each other, then they become less attractive to charges that are already they are screened also by the solvent itself. But now we don't get that dynamic screening. These polar particles are going to be as polar when they're apart as when they are together. So we don't get this dynamic explicit behavior. Okay, so that could be problematic. And in that direction, people have been working in explicitly polar and polarizable extensions to coarse-grained. Mixed success, I'd say. It also becomes more expensive because now we have more particles. More questions? No? I'd say just another question.
That's another one. Around the EuChemS potentials that you use to characterize the interactions between particles through these, are they like come from like a universal lookup table and they're always the same for every model, or can you change the potentials slightly?
Typically, no. Typically, we don't change. Although this new Martini 3 does give you a lot of tools to modify them while still being inside that table. Now you have some flags that can say, okay, this particle is polar but tends to be an hydrogen acceptor, or hydrogen bond acceptor, or hydrogen bond donor. So that was going to modify a bit the behavior with respect to others, right? There are some ideas actually to have a sort of a more continuous interaction table, but to the future. Right now, it's like that.
How does these Martini models work in protein folding or case of conformation of proteins?
It doesn't, right? That's what I said. It doesn't.
Okay.
Yeah, but that was the easy answer, right? So very directly, if you're using this kind of elastic networks, then you're not going to see any folding or unfolding. But I just showed an example where that protein did change tertiary structure and to allow for perform. So tertiary structure is already more dictated by the side chain interactions, and there you have to be lucky. If your system depends on interactions that in a way are well captured by Martini, that could work. That was the case. But sometimes they are also dependent on very specific side chain interactions that are not well captured, and then you're going to have also the elastic net. That said, a lot of new developments have come out in the past year and such, where you have kind of breakable elastic networks that you could actually have a little bit of unfolding or a better representation for intrinsically disordered regions. So people recognize, well, we the developers recognize that that's important, so we are moving in that direction. So, and hopefully at some point we'll have some degree of actual foldability. Let's see.
Just out of curiosity, for the protein that is sensible to the osmotic pressure of the bacteria, is any inhibitor described for that protein? Or?
I'm not sure. Maybe one reason is that we evolved to have the same thing. So you have the same thing in your touch sensors. So when it's not because of osmolarity, it's when you put pressure like this, the same thing happens. So maybe it's not so good to attack that if you have the same. I'm totally hypothesizing here.
Nice. Curiosity, give me any.
Yeah. No, but I don't know. It's called MSCL. Well, you're gonna have a slide if you want to look it up. All right. Well, I said you could have a break, but I don't think we're gonna have a break because then we have to move to the next one. Juanjo, can you help me change the? All right. So because I was told, no, this is not beginner stuff, I'm going to show some more advanced stuff that I was involved in, and that actually shows that parameterization can become quite complex. And we'll focus on cholesterol and sterols in general. And this is something that goes way more than 10 years ago. And okay, so this is the original Martini cholesterol model. So this is how it looks optimistically. This is how it looked in the Martini paper in 2007. Problem is, I mean, whoever has worked with GROMACS probably has seen something like this. Yeah. Links warnings. Yeah. By the way, Link's warnings are almost never the culprit, okay? It's like saying that, oh, the cause of death is cardiorespiratory failure. Sure, everyone just goes into cardiorespiratory failure, but maybe the cause of death was the piano that fell. But no, it's Link's warning. People blame the Link's warnings. Anyway, when people started running long simulations with lots of cholesterols, these things started cropping up. So, but why did they happen? So cholesterol is this molecule that has lots of particles, lots of beads on top of each other. And if you look at the structure, okay, sure, you have this tail here that will have some flexibility, but this part at the coarse grain level is going to be pretty rigid, okay? It's not going to have a lot of flexibility. And the way it was parameterized initially, it was a bit like this. So this is that flexible tail. Sure, I'm representing there with a light bond. But then here, everything was either constraints, what was these white bonds, or these very stiff bonds. So the molecule is a bit rigidified by all these bonds. And then what happened is when you simulate this in a lipid membrane, something like this would happen. So what you have in the background are lipid tails, and here is a cholesterol molecule. And I want you to pay attention to the fact, like the time scales of the movement of things. Okay? This is a simulation that I saved every frame. And by the way, I totally recommend if you're developing force fields and you're hitting problems, definitely do this. Like save a trajectory every frame until you get a crash. And you can see that lipids are slow, but the cholesterol is really fighting its demons. And at some point, it's pow. All right. So yeah, okay, Link's warnings. No, the problem is that this was unstably parameterized. Okay, that was a piano. Okay, so back in 2012 or 13, then I had the idea of maybe using virtual interaction sites. So what is this? This is the kind of the poor man's solution to rigid body simulations. So what are rigid body simulations? Rigid body simulation would be a simulation where you have a set of particles or even shape that always keeps its shape so that it will move according, always with a absolutely rigid shape. And then you'd have to come in, take into account moments of inertia and such. So typically you would not get that in MD engines. Some do. GROMACS doesn't exactly. GROMACS has the virtual interaction sites. They were actually made to simplify the representation of amino acids. So that, for instance, for the tryptophan, you could have like a triangle here. This would be a triangle of constraints. And then all these atoms would be constructed as a sort of a linear combination of this vector and this vector, okay? So based on the position of these three atoms, you can construct the rest of this side chain. Okay? So there's different ways to construct these virtual sites. And the idea is then for cholesterol, instead of having this highly constrained structure, so essentially we want to have something that is fixed in space, but constraints only work in 1D. And I want to do something that is 3D. So then I could use this type and this type of virtual site. This allows me to build a fourth atom in plane with the constructing atoms, and this one is a fourth atom out of plane. I could do something like this. I could, instead of having this whole set of constraints and bonds, I would just have these three atoms. That would be a frame of constraints. And all the other ones, this one, this one, this one, this one, and even this one, they would be virtual sites. So whenever this frame moves, all those atoms move rigidly with it. When one of those atoms feels a force, it transmits the force back to the frame. The frame moves, and the atoms are rebuilt. Okay. So this was actually quite successful, and this is just an example showing that it behaved just the same as the non-virtual site cholesterol. So the distributions of orientations and such were the same. So yeah, I was very happy. But then I had this colleague, the Clément Arnares, this French guy. He's super cool, and this shows how old this work is, because he's by now he's finished his PhD, done a couple of postdocs, and he's been out of academia in data science for almost like 8 years or so. So this is old work, but I'm going to connect with recent work, don't worry. But yeah, so he's working with these huge systems, and for him, it still crashed. So we figured out it might have to do with that maybe the fact it's too rigid somehow is a problem. So we introduced this kind of a shock absorbing hinge. So instead of having a single frame, we had two triangular frames connected by distortion that will allow a little bit of a nyik, and hopefully that would kind of absorb any instabilities. It seemed to work, all right? And then we got this paper out. This is from 2014 or so. And more recently, 2 years ago, we did the same thing now for Martini 3. But Martinez 3, we were able to do it with just a single frame. So turns out we just needed the frame not to be so narrow. What's happening here is that in this approach, the virtual sites don't have mass. The mass is only on the triangle. And if the triangle is very narrow, you can see that you're going to have your center of mass and your moment of inertia around this torsion. Yeah, it's going to be super easy to the moment is going to be very low, so it's going to be super easy to rotate here. So any force here is going to make the molecule spin super fast, and then you're again in one of those problems where the particles just crash against the wall. But now by having a broader frame, this works better, and now the moment of inertia is larger in that around that axis, and everything seems to be fine so far. It's been 2 years and it's working well. So yeah, that was it. Incidentally, the reason why we can now have broader frames, because I told you that now in Martini 3, we are trying to match better the shape. And that means actually placing the particles a bit more further apart. So that worked incidentally very well for cholesterol. Very good. So this was the beginning of introducing virtual sites to coarse graining. I mean, it had been used before, but in Martini, this was the beginning, like this initial cholesterol model. Since then, there have been some problems associated with it. So are they a Poisson solution? And the fact that we had this coupled frame was a bit problematic, not so much because of the idea itself, is that the solver in GROMACS that solves those constraints, especially when you have this coupled frame, made some inaccuracies, had some accumulating inaccuracies, and it turned out that it behaved like a heat sink. So the cholesterol will kind of suck heat out of the system. And if you have one cholesterol here or there, it's not a big problem. But if you were studying that effect that I showed you of the membrane separating, the phase is separating, you're going to have a phase that has a lot of cholesterol. It already likes to be more ordered, and now you're cooling it. So you're really driving it more what you want to see. So it kind of biases the observation. So this paper deals with that. We solve it 2 ways. One is by asking GROMACS to be more precise in solving the constraints, gets a bit slower, or Martini 3. Now with Martini 3, the cholesterol doesn't have this coupled frame, so it works fine, right? This is again one of the examples of the problems that don't happen with atomistic simulations, because that kind of construction of 2 triangles next to one another with constraints, I mean, it doesn't really show up in atomistic molecules, right? All right. Oh, you're doing right. So I think we're going to finish in time. So this whole introduction about cholesterol, so I can bring you to this more recent work on steroid hormones. So I had this collaboration with the University of Santa Catarina, University of Federal Santa Catarina. And so they're interested in studying these several kinds of hormones, in their case for studying their ecotoxicology. Honestly, I'm more interested in the parameterization. That's what I find cool. So whatever they like, yeah, great. So we have these hormones, and you can tell these are very much related to cholesterol. There's a lot of common parts there, and we're going to be looking into that. So this is some part that's cut there is the cyclopentano-peridophenanthrene ring structure of cholesterol, and based on that, you have all these modifications. So the color scheme tries to highlight what is common between all the molecules, and also what is different. So okay. What's the strategy here? We're going to obtain atomistic simulations for each of these cases. Are there even atomistic parameters for all these hormones? Or if not, are we going to do QM? It's a question mark there. But then after we have our reference data, we're going to map to coarse grain, and we're going to compare to the cholesterol topology, to the cholesterol model, to see which particles stay the same, which particles need changing. That seems like a good strategy. But then we thought much better. Now scratch this. At least scratch the top one. Why go atomistic or QM when actually these hormones are super studied in the PDB, because there's tons of structure of these things with proteins in the protein data bank. So we can just use that. So we're using the PDB not for proteins, but for the ligands, right? So once you have that, okay, yeah, then we're going to try to align all to a common skeleton. So those guys are all different, but they all have this this part here in common. So we're going to align that. And after aligning that, okay, we map to coarse grain, compare to cholesterol, and refine what needs to be refined. Okay, so this was the overall strategy. Now, you might be wondering, okay, but how many structures do we actually have in the PDB that we can use? So this slide here shows how many structures are in the PDB for each of these hormones. So for instance, I mean, there's guys like progesterone, 50 something. I think this is the record one. So there's lots of data, but then there's guys that, for instance, like prednisone only have one. So is one enough to model this? And the thing is, because there's this repeating behavior of some consistent moieties, I'm not going to be using only this one to parameterize this molecule. No, this moiety here that is present in the corticosteroids exists in this one, 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 this one. And in total, there's, I mean, 50 or 60 examples of that particular moiety in the PDB. So I'm going to use that information from all of these guys to decide how it behaves in this case, right? Same thing for this moiety, this moiety. There's even this case here. So there's no examples of this hormone in the PDB, but I'll be able to work with it because, yeah, I'll infer the behavior of this one from everyone else. The same thing with this OH there, the same thing with this carbonyl there, all right? So this is a potentially powerful approach, right? So then this is what they would end up looking like, just because they're cute. But okay, so I want to give an example of how we deal with one of these beads, how we decide how to keep or not their parameters. Okay, so I'm highlighting here this bead. This is different from that one in cholesterol because maybe you can see that this one has a double bond here, which is the same one here, same one here. These are all the same here. But then when you have the oxidation from this guy to this guy, the double bond moves to here, which means that the local environment of this bead is going to be different. Possibly the structural placement relative to the rest of the skeleton is going to be different. So we wanted to know how different it is. And if it is different, then how do we translate cholesterol's parameters to it? And so what we do is the following. So this is pretty abstract, but I'll guide you through this. What you're seeing in this light gray is the stick representation of cholesterol atomistic, just from the side. We're just looking at it from the side. Okay, this would be the tail there, and this is all the rings, and this is where that bead would be placed, and that this is the direction of the hydroxy or carbonyl group. All right. And what I'm overlaid here in dark gray and orange is where the cholesterol beads actually are. Okay, so there's a bead there, there's a bead there, there's a bead there. Actually, these are the three beads that make the frame for constraints. Very nice. Now, when I went to the PDB and took all those structures of those hormones that had a different setting here because they have the carbonyl, so they have a different local bonded possibly a local different structure here. What I did is I first aligned those structures all to the cholesterol. So to this common part here that involves it's this part here, this ring, this yeah. So we made that alignment, and then we mapped it to coarse grain. So I'm not showing all those mapped beads. I'm just showing the mapped beads that correspond to that particular one that I was trying to parameterize. And for each of those examples in the previous slide, there is one hit here that ends up in this group. So what I'm showing here is the spread of how those mapped beads end up relative to my cholesterol. So the mapped corresponding beads in the different hormones. And what I can see is that, well, it's close to the cholesterol behavior, but there's a bit of a difference. Most of them, they are around here. There's a difference in the center, and there's also a bit of a spread. So now, based on the spread and on this difference, I can decide, do I just use the same parameters or do I change, right? And the idea here is, well, I found that this was a bit too much. So anything above 0.3 angstrom, we decided, okay, this is too much. We're going to do different parameters for this. And if the spread is too high, in this case, we didn't decide it was too high, but if it's too high, it means that this group has a lot of flexibility. So it's not going to be a virtual site anymore. It's going to be something that has a bond and an angle potential to allow it to do this, all right? So this is summarized here. 2 minutes, okay. So okay, so now it's the time to validate this. So we did this for all the hormones, and then we passed this through this log P simulations. And the trend is correct. So the more apolar ones, they do have a more apolar log P, simulated log P. There is a group of here that starts diverging a bit more to even more excessive log Ps. We're not exactly sure why yet, although this is already at very high apolarity levels. So I wouldn't say this is very critical, but I would like to understand better why this is. But then the cool thing is, okay, we have all these structures that came from the PDB because they're bound to proteins. So can these models actually recover that? So, okay, we have the PDB structure. We take the hormone, but take it out of the binding pocket, just put it next to it in the simulation box, and there it goes. And hey, no, that's not the binding site, that's just a decoy, wait. And goes around, and yep, that's not the binding site either. Some points. Yep. Now it finds the binding site. Actually, the cavity is actually quite in there, and actually finds it, and it's super cool. So we're quite happy. And the thing is that we had all these hormones that we parameterized. So oh, sorry, before that, this is the movie you just saw corresponds to this part here. And what we're measuring is the RMSD of our ligand to the same ligand, but directly from the PDB. So compared to this target when it's in, so we can see that it really reaches the binding site. And well, the binding site in this case is a bit big and it can wobble around there. That's why you have quite some quite high RMSDs even in the binding site, but it was able to get very close to the experimental configuration. And then we did this for all of them, and for most of them, like 80-something%, so all these guys, they actually were able to find the binding site. Only these four didn't. These ones actually involve heme cofactors, so we think that the heme might have some problem, but well, to be decided. But turns out that this seems to be quite good. And that's it. So this is the conclusion of this quite extreme parameterization based on experimental data. So who needs final resolutions when you have the PDB? Screw atomistic MD, right? There's still some excessive hydrophobicity. Well, we have to look into that. We had a very good overall recovery of the binding sites, but the heme might need some more work. And I didn't talk about this, but there's some degree of protein model dependence. And the thing here is that some of these bindings are induced fit, or if you want to be more intuitive, some of these binding pockets are kind of hidden and need a bit of unfolding for the ligand to come in. And if we have a very stiff protein model or elastic network, this is going to be problematic. So we tried several models. We tried like 4 different protein models, and we were happy if the ligand found its target in any of these models. So maybe one of the models was not going to be enough for all the hormones, but with all those hormones, at least with one of those models, it worked. So I'm happy with the hormones. That's what I'm parameterizing. If there's problems, yeah, that's the protein site. And that's it. So a big thanks to Tamala, who is the PhD student who did all this work. And that's it. Questions?
Manuel, if you agree, but if they have questions, they can sit with you during the live.
Oh, perfect.
Because we only have 1 hour and 15
No,
minutes.
that's great. That's great. So let's do that. Yep.